Thank you everybody for attending for this special webinar sponsored by IVF 2.0. So the principal aims of this session is to look at the state of play for the crossover of automation, robotics and augmented reality in assisted conception. So we all recognise the importance of staying at the forefront of the scientific and technological developments and their potential to revolutionise the field that we work in. So this webinar is a proactive approach to push the boundaries of reproductive medicine and explore the full potential of this new discipline, such as automation of the IVF lab plus robotics. And by leveraging artificial intelligence, microfluidic devices, self-contained systems, laboratories can achieve increased efficiency, precision and reproducibility in crucial procedure steps such as fertilization, embryo culture and cryopreservation. So this advancement may not only simplify complex processes involved in IVF, but has the potential really to democratize access to IVF by reducing costs, improving outcome, and addressing maldistribution of resources. We'll keep the bios short and you can read all about the participants on the main website, but I'd like to briefly introduce my two co-hosts. Stephanie Kuku wears several hats. She's a trained gynae-oncologist, trustee of the King's Fund, honorary senior research fellow at UCL into artificial intelligence, and is also an advisor and investor. And her latest venture is executive vice president of scientific affairs at Conceivable Science Life Sciences. So welcome, Stephanie.
Thank you.
And then Matt Pettit is chief scientific officer, general manager at IMT Matcher, posted his whole for the last 16 years. And this company provides cryopreservation systems in animal breeding and electronic witnessing and QMS for IVF clinics. So welcome, Stephanie and Matt.
Thanks. Thank you very much, Andrew. Please allow me to introduce you as well. Probably many of the people listening already know you because you're world famous, but for those who don't, Dr. Andrew Drakeley is one of the co-founders of the IVF 2.0 and an honorary clinical associate professor at the University of Liverpool. And Andrew is also a consultant gynecologist at the Hewitt Fertility Centre, which is also in Liverpool. So thanks very much for the intro, Andrew. As someone who's already directly involved with the development of new and innovative ART that are being designed to help streamline efficiencies in the practice of IVF, and perhaps more importantly, to mitigate risk, promote best practice, and ultimately to assist with the patient's treatment, I can really appreciate and fully identify with the comments that you've made. So thanks very much, Andrew.
Thank you, Matt.
Thank you, Matt. Thank you, Andrew. It's a pleasure to be here with both of you. We are going to get started. I'm delighted to be here. It's such an exciting time for IVF and automation. Two reminders: certificates are posted in a few days from now. And tonight, please ask those questions in the Q&A section and not in the chat section. So I'd like to go straight ahead and introduce the one and only Denny Sakkas, who's got a talk for us, the Chief Scientific Officer at Boston IVF and consultant to a number of clinics and startups in the area of IVF. He's also adjunct professor.
Welcome to the latest i3 initiative, and thank you to the i3 team for this invitation. And today I'm going to talk about modernizing embryo handling and culture. My name is Denny Sakkas, and I'm the CSO at Boston IVF. So we're all well aware that we can grow these embryos in vitro, and we can see them. We get amazing images from time lapse. But what we really, understand is that there's a lot going on behind just watching this embryo for 5, 6, or 7 days after fertilization. And we've become very good at culturing embryos. But obviously, as I said, there's much more going on that we have to really cater for. And we're very lucky that the embryos, behave in the culture media we've produced. And one of the things we know is that, for example, maternal messenger RNA drives most of the embryo development up until the cleavage stage or the compaction stage and the 4 to 8 cell stage in the human. And then once the embryo is actually using its own transcription, the embryonic genome is activated, and from the 8 cell to the blastocyst stage, we get these amazing gene transcription from the embryo itself. There's much more that actually goes on behind the scenes of what you're looking down in your time lapse or when you're looking down an embryo also. And one of the biggest things we've learned over many, many years, and a lot of this data came from studies in the 60s, and then the great studies by Henry Leese and colleagues, where we found out that there's this schizophrenic switch in how the embryo metabolizes, and it goes from this switch in the early stages where it requires pyruvate and lactate, and then it switches post-compaction to glucose. And this switch in metabolism is what actually probably changed the way we look at embryo culture media, and how we change the culture media from the very simple 70s and 80s culture media that we used, which was basically a set of 7 or 6 ions plus some metabolites, to what we currently use today. So the way we modified embryo culture is actually quite fascinating. And we went from these simple salt solutions with these obviously added energy substrates that catered for this metabolic shift. Some people use complex tissue culture that we sort of stole from other areas of cell culture, which were really not specific for embryos. And then we had this change where the great studies that David Gardner did, John Biggers and others, many others did, we changed over to these sequential media, which sort of catered for this is what I like in the fallopian tube for the embryo, to this is what I like in the uterus. And then through studies by John Biggers and colleagues, we changed the culture media, and we got to these one-step media. And we're still, there are still some labs that use sequential media that like the idea and the concept of, I want to cater how the embryo's requirements are during different stages, to I just like to, keep a homogeneous environment for the embryo and keep it in a one simple media. And they all work very well, and we get great pregnancies and great blastocyst rates. But there's much more to culture media. Obviously, we've got the one step and the sequential media. There's the type of environment we grow them in. We just still grow them in a little droplet, and that's still, something we really haven't quite understood. And so the little droplet under oil is something very foreign to an embryo, if you want, because really the embryo in its fallopian tube, in its little crypts in the uterus, is growing in a very viscous environment, and we haven't really even looked at viscosity in terms of how that may impact embryo culture. We used to do a thing called co-culture, where we used to grow embryos on feeder cells to try to mimic the fallopian tube or the uterine environment. And again, this is something we haven't really looked at. And then there's the gas phases. We've actually become very good now at understanding much better that the embryo really requires lower oxygen environments, and hopefully everyone listening here is culturing in a lower oxygen environment. But should we even go lower? Should we cater for this change in the uterus where it goes from sort of a higher to a lower oxygen environment? And obviously, do we need new types of incubators? We've got very good at these benchtop incubators. We've got the time-lapse incubators, but should we be looking at new types of incubators also? So there's a lot of things that we can look at to change in embryo culture. And obviously the components of the embryo culture media, there are things we can look at. Are we using the right macromolecules? Should we add in antioxidants? There's some great work by David Gardner looking at adding antioxidants. Again, cytokines, growth factors, which I'm not that great happy about, but these are things we should all be looking at. And do we have the right amount of carbohydrates or metabolites? So these are all questions that we should be questioning or looking into for new culture media. But is there more than just the metabolic shift? So I'm just going to show you some data that we've had recently to indicate that, maybe we're not really catering for everything the embryo needs in this environment we're giving it. Can we learn more about the metabolic signature of a human blastocyst, for example? So we've been studying for quite a while now the type of metabolism that an embryo undergoes and the metabolic shifts using a technique called fluorescence lifetime imaging microscopy. And this is just looking at two factors, NADH and FAD, which are both linked to mitochondrial function. But we've been lucky because we've learned quite a lot of things. And if you're interested more in this, you can look up some of the papers. The top paper I'm going to talk about a bit more about from Marta Venturis, looking at the metabolic state of human blastocysts. And then there is some data that we've got on differences between euploid and human an aneuploid human blastocyst. But what have we learned about human blastocysts? So we've looked at over 215 human blastocysts, and what we do is we scan through them, and we've got this type of data, this metabolic data, and I'm going to quickly go through some of the key things that we've learned about embryos when they're in culture media. And all this is published by Venturis in 2022 in Human Reproduction. So one of the first things we learned, which is quite fascinating, is that human blastocysts show distinct metabolic signatures between the inner cell mass and the trophectoderm. And you can see here on the left, the blue dots are the inner cell mass and the orange dots are trophectoderm. And what we can do is basically draw a line between some of the parameters that we see in mitochondrial function between the inner cell mass and trophectoderm. So the inner cell mass and trophectoderm cells have very different metabolic states. What about zona pellucida? We look at the outside and the inside trophectoderm of the hatching embryo. We see that the outside and the inside trophectoderm also have very different metabolic states, and these are very evident in terms if you look at the FLIM data that we get from these two types of components of the embryo. So there are distinct metabolic states of the trophectoderm inside and outside of the zona. So the hatching process leads to drastic metabolic changes. What about the metabolic state in relation to its morphology, to its Gardner score? So are we getting more information from an embryo's morphology score in relation to its morphology and its metabolic state? So what we did, we plotted various metabolic parameters in relation to the grading of an embryo, and this is the Gardner grading, AAs, ABs, BBs, and others. And when we looked at it, we actually saw there were really no significant differences. So really, the blastocyst morphology had no impact on its metabolic state. So an AA or a BB may have had exactly the same metabolic state, and two AAs may have very different metabolic states, which is interesting because embryo metabolic state and morphology could then offer synergistic information, maybe in the future for embryo selection. The other thing that really intrigued us is looking at embryos as they progress from day 5 to day 6. And one of the things we found is that, again, we actually saw that day 5 to day 6 parameters in embryo mitochondrial state were very different. There's like an internal clock in the embryo that tells it metabolically it's at day 5 and tells it metabolically it's at day 6. And that's regardless of morphology again. And finally, we have some intriguing data indicating that mitochondrial status or its state could be related to the ploidy status. So euploid and aneuploid embryos may have very different mitochondrial status. So, and finally, if you're really a metabolism geek, you should read this paper by David Gardner showing that lactate production by the mammalian blastocyst is actually in manipulating its microenvironment for uterine implantation invasion. So the metabolism of an embryo is very key to its status and how it grows. But let's go back and say, what do we know? We know that the blastocyst utilizes high levels of glucose. It has a high oxygen consumption, and it converts around half of the glucose consumed to lactate by a process called aerobic glycolysis. But it's much smarter than just that. It has a distinct metabolic signature in the ICM and trophectoderm. It has distinct metabolic states inside and outside the zona pellucida when it's hatching. It has its metabolic state is really not associated with its morphology. It has a metabolic state that changes by day 5 and day 6, in particular as it expands and hatches. And the metabolic state may be associated with its ploidy status, and it creates its own metabolic environment during implantation. So why am I talking about this? Why am I interested in actually understanding the metabolism of the embryo and understanding other key factors? Well, in the future, I think all these factors will help us to actually change the way we actually culture embryos and look at the way we culture embryos, because I think we can bring in novel technologies, automation, probably artificial intelligence, and in particular microfluidics, to change the way we actually accommodate the growth of the embryo. Obviously, we can't really mimic entirely the way the embryo grows in vitro, but I think we can change the way things happen. And this will be through various new technologies and automation in the lab. This is a lab on the chip technology, and this was published in 2008 by Gardner and colleagues here in Boston. This is a very kludgy looking system, but basically what it does, it actually has inlet and outlet valves which measure the way the embryo is growing in each individual droplet, and actually can take out the media and assay it virtually in real time, hopefully. So this is a way of growing embryos microfluidically and allowing analysis of single pre-implantation embryos. These are other systems, again, developed by Jeremy Thompson. This is actually a system which cradles the embryo in a microfluidic device. This type of system was allowing it to actually be micro-injected using like this cradle to hold it in place while you're injecting it. But you can see that the same cradle could be used to actually hold the embryo in place while you're shuttling in culture media and actually taking out culture media to assess it. And this has been really well described by Gary Smith and colleagues who've been doing this for many, many years. And this is a great review showing how microfluidics may allow real-time morphokinetics, may allow real-time metabolism assessment, and even non-invasive PGTA. So you can picture an embryo developing in these microfluidic chambers and growing and being analysed and having the media sort of being changed as it's growing. So this will all depend on new media that we develop and how we develop those new media to cater for embryos at different stages. So how could this all sort of be put together in a future lab? So what we know about is there are techniques now to allow us to do sperm injection, to do sperm sorting, microfluidics to pick the best sperm. There are also artificial intelligence and imaging software that allows us to pick the best sperm. There are ways to collect the oocytes and denude them now that are being discussed. There are definitely ways where we can inseminate or automate the fertilization process. And then what I've been speaking about is how we can actually automate and culture embryos in a microfluidic environment and then pick which ones will actually be frozen or transferred. Now, all this will be driven by artificial intelligence, but not just artificial intelligence aligned from embryo images, but actually bringing in metadata, patient data, knowing patient age, patient characteristics, and also bringing in analysis, real-time analysis that we can get from the media and from aspects or metabolites from DNA and other components of the media that will tell us all together which embryos and maybe which ranking order these embryos will be frozen or transferred. So in the future lab, we'll have all these capabilities, hopefully, that will allow us to culture embryos in a better state and analyze them in real time and know which ones we should transfer. So how will this hopefully work in the future? So envisage a microfluidic chamber which individual embryos, and each embryo will actually have media coming in and media coming out. And it'll be like a personalized embryo culture media. So the system will know what stage the embryo is and bring in media that is actually relevant for that stage, whether it's the blastocyst, whether it's a two-cell fertilization. So we'll have a personalized embryo culture system that will actually cater for the embryo's needs at that particular stage. And it may not be a continual liquid environment. It may be a state of more viscous environment, maybe changing viscosity as the embryo develops. So as this goes on, we'll also be analyzing the embryo, and hopefully we'll have technologies that will actually allow us to know which embryos to freeze and actually assign an order to them to rank them in order. So hopefully this is the way we will cater for embryos in the future as we learn much more about the pre-implantation embryo development needs and stages and the culture protocols that we should follow. So thank you. If you have any questions, I'm always happy to answer any emails. And again, thank you to i3 for allowing me to talk today. So obviously the question is, when will we have these technologies in our laboratory? And the good part is that, you know, we have microfluidic systems in our hands now that may allow us to start doing the embryo culture. So we saw the little cradle system by Jeremy Thompson and David Gardner and colleagues, but we also saw that the 2008 paper of a microfluidic system of analysis, and that's been 15 years now, and we haven't got it in our lab. So these things will come as stepwise. I think we can confidently say that in several years we may have a microfluidic system that allows us to culture embryos and probably do some rudimentary analysis to flush out the media and get an analysis of that, whether it's through metabolites, through DNA, RNAs. But then I think slowly we will probably allow us to actually culture the embryos and actually bring in media of different types, maybe initially like a sequential type media of a stage 1 and stage 2. But I think that microfluidics will allow us to expand on that and give different aspects of media during different stages. I think we're still far away from the final footprint where we can do personalized embryo culture if you want. But I think the microfluidics is much closer. I think changing and doing some rudimentary analysis is probably the second step. So I would envisage in 10 years' time, we will have some sort of microfluidic system that we're culturing embryos in.
Well, that was a great first talk from Denny. He's not here with us today because he's traveling en route to Esri in Copenhagen at the moment. But a couple of things sort of spring to mind listening to his talk. We've seen a couple of papers on abstracts, mainly in conference abstracts at this stage, about automated denudation of oocytes from clinics in USA and China. But it's really, you've got to look at some of the simple stuff that takes embryologists a long time, like simple dish prep, for example. And it's the automation of those simple tasks that would save the embryologist a lot of time that I think are equally as useful. And maybe it's time to look outside the field of medicine, even aeronautical industry. NASA, has interest in these sort of areas. So there's a lot of scope for development there. Okay, so I think we'll move on, and please put your questions in the Q&A, and we'll come back to Denny's talk as well at the end, and we can then discuss that as a group panel. That'd be great. So our next talk, it's my pleasure to introduce for the first time to i3, Dr. Christine Allen. She is a certified embryologist and andrologist who's traveled the world globally, teaching leading-edge vitrification and automated lab technologies, building IVF clinics and implementing new technologies such as state-of-the-art egg banks, not just in the USA, but South America and other countries as well. So thank you, Christine. And Christine's going to talk about vitrification of oocytes and embryos.
Thank you. Hi, everyone. Thank you for being here, for listening to us. So let's talk a little bit about what's out there clinically available and what is in research phase, what is beginning, what is not, on specifically the automation of vitrification of oocytes and embryos. I'm Christine Allen. I'm the CSO of North Star Fertility Partners. I'm also the COO of Elite IVF Consultancy in USA, which just merged with Fertility Consultancy in Denmark. I'm also the fertility advisor for CryoFuture, CryoStorage in US, and I don't have any conflicts of interest to disclose at this time for this specific subject for this presentation. So welcome, everyone. If we think of a timeline of how we're handling and using cryopreservation in IVF, we see that just 40 years ago here, we had the very first success for pregnancy from a cryopreserved embryo. And not that much improvement was showing until we got to the 2000s when we adopted vitrification instead of just freezing or slow freezing of embryos, right? We got great improvements from our rates and our success from vitrifying, but we did that at the cost of some lower reproducibility and introducing an open system to cryopreserve and to cryostore our material, which has the potential, and I know this is a big debate, but it has the potential to open the door for certain disease spreading. We fast forward another 20 years, and it seems to be 20 and 20 years waves, right? But we get now to 2020s where we are working a lot on automate this process of freezing. By automating it, we, at the same time, address and mitigate the problems that we actually created by introducing vitrification with less reproducibility and open system in our field. And this is one of our goals here, to address and mitigate the problems that came with the technique. And everybody always asks me, okay, I don't see studies about automating vitrification. What is out there? What? It's nowhere. Well, it's not nowhere, it's there. We just have to open and widen our viewpoint. These studies may not be published in the 3 or 4 periodicals that we read from embryology and reproductive endocrinology from our 2 or 3 societies that we follow, but they are out there in quite sophisticated periodicals for electronic engineering and computer science. And of course, I'm biased because I have my crew that works with this in here, and I have access to this, but access is free for everyone. So if you really want to educate yourself on what's coming up, what's going to hit our labs, and how this works, there are some other periodicals like the IEEE Explorer that is going to be very helpful for you. Another question, okay, it's not even in our field, yet I don't see it anywhere. Why do I have to worry about learning about automation right now? Is there any evidence that I should be worried about this, that I should be learning about this? Actually, yes, evidence is very clear from all the studies that are out there. There are 2 main primary benefits from automating vitrification. The number 1 benefit that hits everyone is the elimination of unperceived human error and elimination of variability on our results. What happens here is when we make a mistake, freezing or vitrifying an egg or an embryo, we don't see that we made that mistake. We have no idea as humans that we made that mistake. So that's going to be seen in the translation into a huge variability on the success rates from one technician to the other, one day to the other, morning to afternoon. And that's what we're trying to tackle here. We want precision, we want accuracy, and those are being achieved with the automation of the process. Second benefit that I'm biased, again, because I am at a high-level management of a cryo egg bank, is standardization of procedures. Whoever is watching here that manages many labs or manages cryobanks, the production of viable oocytes to go to recipients to produce pregnancies. Standardization of protocols is our nightmare. You know what I'm talking about. So with automation, of the system of at least the vitrification part, putting those eggs in storage, you already have a huge improvement in your results, or at least you should see those results much better. But everything comes with disadvantages, right? And what we see in here from all these studies that are out there, and even my own experience in here, the disadvantage today to automate anything, not only vitrification, is exposure to cybersecurity threats, which is a major problem for health sciences and medicine, healthcare these days, and tech support. If we're going to use this in an IVF lab, we need perfect tech support. We need real-time monitoring systems. And I always talk about the difference between real-time monitoring systems and data logging monitoring systems. For this type of automation, it's very hard to work with data logging because everything has to be very fast. We need to be, as embryologists, the tech support inside the lab. We have to understand what's inside that program, what's inside that machine, and how to overturn it, how to take over, how to troubleshoot it. I like very much this table. It's brilliant. I wish I put this together, but it wasn't me. This is a courtesy of Dr. Taya Peura from Australia. She's brilliant overall as a researcher and a manager. But this in here, you see step by step, everything that can be improved by automating in a compare-contrast between manual vitrification, the artesian method, and automated vitrification. I'm not going to go one by one in here. I'll leave it for you guys to read this later. But it's brilliant and it's very enlightening if we want to learn more of why we're doing this. Okay, so now let's take a look on what's out there. We have a supporting publication on, first of all, the concept of the robotic arm. And I know you think of a robotic arm and you think that giant metal arm, right? That has those joints that make noise and the little fingers in there. No, this is just this came out of that concept. You don't have those arms grabbing you around in the lab, obviously. We have one example of that concept applied to vitrification that uses, again, as Denny Sakkas was saying, microfluids. It's microfluids plus electrodynamic forces that move the embryo or the egg back and forth in there. This has been published years ago. It never went in the market. And you can see the literature on each one of them in here. I added for you guys, anyone that wants to read more about this, please take a look at the literature. This is 10 years old. It's out there for a long time, no market. We also have the attempt of using automatic pipetting, like you see in virology or in blood analysis. So it just pipettes like your hand for you, from here to there, from there to here, until it's the end. Here we have a picture of a commercial machine that also hasn't been commercialized much for the past few years, but it showed up in the market at ASHRI a few years ago. And we have another interesting one in here that is an entire gadget. The entire microscope moves for you. And this is also 10 years old, was patented in US and still not commercialized. But it's out there, it exists somehow. And this is the beginning of the evolution that we have on automating things. Another approach that is completely different than any of this, and we have to take a breath and open our minds to understand how this can work, is the semi-automated technology. This one is already available for clinical use. It's in the market. Some countries already have a release for this to be used as a clinical tool. Some others shall not. And in here is where you're going to find the majority of the data, the evidences, and the clinical trials on how this works. This all these studies that I put in here, and there are much more on this approach, they have shown a diminished interoperator variability. Some words I just can't say, sorry guys. So not much difference from tech to tech. It's time savings and it can increase our clinical outcomes. It's everything we want. We want to save time. We don't want a variability between our embryologists, and we want better clinical outcomes. Lots of studies on this approach, particularly. This picture in here in the middle is from a commercial website. I'm not going to say their names, but if anyone wants to ask me which one is clinically available out there, we can talk about this later. It's organic that if we are working on several different tools that will automate different things in the IVF lab, we have to think about chips and pods that will merge those technologies in a streamline, right? And the beginning of that, we can study a little bit about with the work of Wang in 2018. This is a very well-written piece of writing, of course. It's a very well-written study that shows that automation can really work well. And it concluded that it's a viable option for us to be using in the labs right now, merging with other automations. So it's compatible with a lot of other things that we can do in the lab. I put in here two other examples of automation systems for vitrification. One is more academic, and you can read more the intrinsics of how it works. And another example I put in here is called the Embryology Lab in a Box. This, to me, is a mystery because it came all in the market around 2 years ago as a big novelty, but we don't have much information about it. We don't know how it works inside. We don't know what is the concept. Is it a mix of concepts? It's all intellectual property. To me, it's very hard to accept something that I don't know how it works inside the lab. I want to know how to troubleshoot. I want to know how to make it better. I want to know what to expect in the moment of a failure. So this came really hot with a lot of market and then disappeared, at least to my view. If it's out there, I don't know. I here recommend the reading of Wang and all 2018 that can clarify a lot of questions we might have about this. And full integration, it's where we're heading to, right? Mainstreaming, putting everything that we're talking to each other today in here, how to put all our work together in one tool that is going to make everything automated. It's the organic growth of how our field is evolving. We have to either accept it and work with it or let it go. It's a Darwin evolution theory. Abdullah this year conducted a study more specifically on oocyte vitrification that was very enlightening on this matters. And I recommend you guys read it to learn more into this. The study did concluded that automation of oocyte vitrification will reduce cost, and you have good calculations, easy to understand in there, and obviously improves safety. The study also found that automation can reduce the time needed for oocyte vitrification. Everybody wants that, right? We want to reduce costs. We want to reduce time and have a safer procedure. So I recommend this paper too. All these bench tops that are under development, and this is something completely new, of course, and it's not out there for us to test and see how it works. But anyway, the research is being done. They are being designed to be very user-friendly, and they are going to be workstations that have all the advances with safety features. So everything is going to be in your workstation. You don't have to learn and buy from different vendors 100 different things and try to make them converse. No, they're going to offer you one solution that has everything on it. And to me, even though this is just starting to be under development, to me, this is the silver lining of all the hurdles that we go through, automation attempts in our IVF lab. It's very exciting. I added in here another suggestion of reading that is about robotic systems that mix the microfluids and the statics and the robotic arm, but also a little bit of the semi-automatic approach. When I say robotic, we move embryos or eggs or whatever sample you have in there from place to place. When I talk about semi-automatic approach, I'm talking about moving the medium around the embryo or the egg without ever touching this embryo or egg. And I know it's a different concept. We're not used to this. It's hard to grasp at first, but it's working beautifully. And I think as long as we have the data and the evidence, it's worth to take a look at it. One thing that we see in all these studies, the importance of human oversight. Who's going to design? Who is going to implement with validation? Who is going to supervise all these automated systems? Who is going to work on optimizing them? Who is going to understand the robotics to make sure we can troubleshoot? The normal embryologist, the usual embryologist that we see in the lab today is not going to be the person doing that because we don't have that degree of importance to basic and applied biology of development right now as a requirement, let alone IT training. So I think the big question is, am I going to lose my job? Yes, probably. A number of us will not be in the field in the next 10 years for lack of knowledge. Now, if you are out there posting and talking to your friends as search engines are AI or Google AI, you have to start understanding what you're talking about and what all this means, because somebody that is making the mistake, search engine and AI, and interchanging those terms is probably in the line to not be with us in the next 10 years. Let's look for education in this area. Let's try to understand what's happening, but a little bit deeper to make sure that we're there, we're growing with the field, and we're going to be able to manage all this. Next steps, what's coming next? Okay, we have all this work on automation of the vitrification process, the step 1, that is making our eggs and embryos ready to be cryopreserved and stored. But what about the other side? We're going to need to automate warming too, right? Very little done in this field just yet. It's just starting, but there are some really interesting studies about it. We have one in here that I use as a citation for vitrification warmed with the use of laser to tailor exactly the warming rate for those embryos and eggs. So it's starting to show up, it's out there. It's just not in our periodical. So let's broaden a little bit what we're reading. Concluding here then, guys, we can see all across papers published out there that automation is a powerful tool in oocyte vitrification, embryo too. For me, as a commercial cryobank manager, I can see that in the next 5 years, this is going to be a must for all the oocyte cryobanks. Whoever is going to be doing artisanal work, vitrifying the oocytes, will have a variability in the results that is not going to be acceptable. IVF clinics just follow suit. What we want in here and what we need is standardization of processes, better precision, better accuracy, consistency on our outcomes for pregnancy, and reproducibility in here. We shouldn't have that much variation from one patient population to the next, just because our cryopreservation method is varying too much. And I'm going to stress this just as many times as I can in here, the importance of human expertise alongside automation. We need experts that know that much biology of development and IT to interpret and to troubleshoot everything that is happening as we absorb this in our market. So I hope this short introduction to automation of vitrification left you with a lot more questions, a lot more confused, but at the same time wanting to look for the answers and wanting to get educated on this matter so we all can have the best labs that we can with the best pregnancy rates for all our patients. Thank you. I leave you here with a list of readings, not only specific on automation of vitrification, but on automation of labs as a whole. Again, I'm Christine Allen, and I'll be here to answer whatever questions you guys might have. Thank you.
Christine, thank you very much. Are you there, Christine? Can you hear me? Hello.
Yes, I can hear you.
Thank you, Christine. That was an extraordinary roundup. I think very, very extensive, exhaustive. Thank you for the references, because a few people have asked in the chat about the references. Precision, accuracy, reproducibility, and consistency. And you've talked about systems that have been built with limited, varied evidence. You talked about the mystery system without transparency on its operating principle. A lot of the questions are sort of falling on this my thoughts, which is, if we have had systems that are either partially or fully automated onto the market and they haven't got traction, why is that? And how can we prove that in these systems? How can we prove when we build something, and we know that, you know, it will be done, and, you know, in the future, as you said, vitrification will be automated, but how do we get people to trust these systems? How do we get adoption? How do we prove precision, accuracy, consistency, and reproducibility in order to make sure that these technologies are adopted?
Thank you. I believe this is the most important question. And it's the question that everybody takes them time to understand. The market is resistant. I was in the development of vitrification itself in the end of the 90s, beginning of the 2000s with Kujawa and Smith and Rebecca Gilbert. And we were done with the technology with lots of evidence by 2001. And the market didn't start accepting that vitrification was better than slow freeze until 2009, 2010, because it was requiring a much higher skill, right? Embryologists needed to be skilled to use that to improve their rates. This is happening again. The embryologist doesn't have to be artisanally skilled right now to use automation, but they have to understand IT. They have to understand biology of development enough to get the clues if something is not going well and fix that, to develop the program. Some of those studies, there's something very particular that we call it, the robot didn't get the memo. That is one of those studies you guys are going to see if you read it. The robot places the embryo in the cryo device and completely sucks all the medium from around it. So the embryo is frozen just in liquid nitrogen, not cryopreservation medium. And now we know that this is terrible for the embryo. But that memo came 10 years ago. Everybody knows that. You don't suck the medium out. And I'm going to say it 10 times in here. You don't suck the medium out, because I do that all day long. But you program the robot to do that. And then your results are terrible. With good evidence, with good transparency of your results, your data, and publications that have that opening, I believe we're going to get there. But of course, if we have things in the market that show you nothing, it could be a Pandora box for what I say. Yeah. And once you use it, all the bad in the world are going to come out of it. But transparency, keep doing the good work, and showing people what is going on inside your research, I think is the best thing. And again, the semi-automated approach, and I promise I wouldn't say the name of the company here, has a lot of data out there from clinical trials. And I think it's a very good reference to convince people, okay, that this is going to take over anyway. So let's start with something that gives me the data and the reassurance of the evidence.
Brilliant. Thank you. I think that was a brilliant answer. I'm going to move on. Thank you very much, Christina. For this next talk, this man needs no introduction. He's an accomplished scientist in his own right. He founded Repro Genetics with Jacques Cohen and David Saville, the first commercial US PGT lab. In 2017, he co-founded Overture Life, a company that is focused on automating the IVF lab. It's a great pleasure that we all hear from Santiago Munne.
Thank you. Thank you very much, Stephanie, and thank you very much, I3 team, for inviting me here. Let me put up my presentation. Can you see it?
Yes, we can.
Very good. Well, I'm being invited to talk about automated ICSI, although for just to tell Christine Allen that this actually is not the ICSI device. This is the Vitri device that is our automated system for vitrification. And she can read about it because the patent has been published yesterday. So just a small comment on the previous and great presentation. So some disclosures. I'm the Chief Innovation Officer at Overture Life, and I'm a founder in other companies, and I'm in several boards of advisors. Well, why we want to do automation? I think everybody has talked about this, but basically the demand for infertility treatment is increasing worldwide, but the current IVF model cannot scale as fast. This keeps prices highly unaffordable for many people. In the US, for instance, it's thought that only 10% of the population, only 10% of the population can access IVF treatments. And the process is still mostly manual, producing inconsistent results. This has been discussed in the previous talks. But one thing that has not been discussed is that also by the system that we have right now, it keeps the data in silos within the clinics, and it's not sharing this data. Therefore, it's making new improvements very difficult. For instance, this is the amount of IVF cycles that are being done in the US in the last 10 years. It has doubled. Sorry, I don't know why it goes too fast, more faster than what I'm talking. But the problem is that we don't have enough reproductive endocrinologist trained every year in the US. And also we don't have enough expert embryologists in the US to absorb all this demand. And obviously this results in expensive IVF treatments that only a portion of the population can afford. The second problem that we have is that, as I said, most of what we do is manual, and this produces that variability. For instance, aneuploidy rates in egg donors vary from 20 to 60% depending on the center. Pregnancy rates in egg donors vary 10 to 80% depending on the center. This is data from SART. I mean, you can just compare, although they don't like that you do that. But if you look at pregnancy rates in egg donors per center, it's mind-boggling that you could have from 10 to 80% variability. And most methods and skill levels vary center to center, operator to operator, and day to day and hour by hour on the same operator. Some examples of this variability. As I said, we did a study a long time ago about comparing aneuploidy rates in egg donors between different centers. And what we found out is that although the average aneuploidy rate per center was 31% in egg donors, there was a huge variability from 18% to 60% aneuploidy rate. Obviously, we investigate later different sources of this variability, but it's in every step of the process. This is also very interesting and coincides with what Christine Allen was saying. This is data from a single ovobang freezing in the same lab and sending the eggs to 55 different clinics. And you could see just the survival rate per center varies from 40% to 100%. So huge variability. Another example is trophectoden biopsy. In theory, there are a couple of papers, very good papers, saying that there is no detrimental effect of blastocyst biopsy. But these are from the same center. But if you don't do it correctly, this is the previous centers from Richard Scott and Tiggs et al. Those are very good centers that have tremendous experience in blastocyst biopsy. But if you don't do it correctly, there is also evidence that you could have a reduction in pregnancy rates. For instance, large biopsy size produce a decrease in implantation potential, depending how you do the blastocyst hatching. Also, there are reports that affect that. Artifacts in PGT, if you don't do the biopsy well, you could create false results that will show us as complex abnormals. And there are several papers now, including one of mine, showing that in randomized trials in young patients, we don't see an improvement in results or a decrease. Meanwhile, there is still a 30% aneuploidy rate in young patients, in the blastocysts of young patients. So there could be an effect of the biopsy there. So what's the solution? Obviously, it's automation. And this will provide us with consistency, so same results in every center every time. It will give us labor savings, which is the major cost now in IVF centers. There's a worldwide shortage of expert embryologists. Also convenience, obviously. For instance, there are some companies working on monitoring devices that can be performed at home or in an IVF office instead of going to an IVF center. And infinite data. If all these machines share data between themselves because they are from the same manufacturer, then you can have better design and more experienced teams than just the data that you have within your own center. And then platforms. If you have a baseline because you're using the same machine that works always in the same way, then you can improve the research that you do, because otherwise, if your baseline changes every day, it's going to be very difficult to demonstrate that something works better or worse, right? So we need more platforms like that. So I've been invited to talk about automated ICSI. This has been a collaboration between Overture Live, Embryo Tools, and New Hope. I'm going to show you a video of how it works. But first, just to say that there have been many other, well, a few other attempts at doing this. This is just one of them. And here, what we are automating, it's only the injection. Okay, I'll put now the video. Let's hope it works. Yeah. So we've done many experiments first in several animal models. For instance, hamster model, in which we had the same survival rates as the manual step. We did also rabbit models in which we had the same survival and cleavage rates and blastocyst formation. We use different models because each one have different characteristics. Some have a thicker zona pellucida, others are the same size as human, and so on. The other one was a mouse model in which we also have pups resulting from it, and we have more or less similar results as the manual process. And once we were comfortable that it was working in animals, then we moved to human. First, we started injecting human beads. Human beads. We started injecting beads into human eggs. And here, as you could see, we had the same survival rate as with manual. And then we did a small pilot in the US at New Hope, where we had the first pregnancies. I'll show you for another video. the beauty of the piezo is that there is no resistance from the other side, so you can inject equally well each one of them. So there is not the problem of trying to suck in the membrane of the egg or anything like that. Here it has made a hole, and now it's extruding that piece of zona. I hope you can see it. Now the piezo is not working yet. When it reaches a certain point, that with this, and here the piezo goes on, and it pulses it perfectly every time. Okay. So this was a small pilot that we published. Nuno Costa Borges is the fourth author in the paper, and basically what we did is compare the same eggs from egg donors. We split it in two groups, one manual and one ICSI, and so half of the eggs from each donor were injected in with one procedure. And we had similar fertilization rates and blastulation rates, but we have a slightly lower euploidy rate. I don't know if it's significant, but this we think that it was because basically the system needs to be a little bit faster, and this is, as I said, this was our first attempt in a human situation, and also when we did the pilot, the person manipulating the robot was actually an engineer. It was not an embryologist. So he did the sperm selection, obviously, not as good as an expert embryologist. We didn't want to automate yet the sperm selection because, as you know, there are a bunch of now biochips that can select sperm. So we think that that's the easy part. That obviously can be integrated very easily with this system, and that's the next step once we get the euploidy right. But basically, we had 3 euploid embryos replaced in 2 women, and it resulted in 2 pregnancies and in 2 babies. And this is the first baby that we have with the system. And here is John Zhang. And that's it. Thank you very much, and I look forward to your questions. If you have any question that you want to write to me, here is my email.
So thanks very much, Santi. That was a brilliant talk. One short question for now, and then we'll take more from the Q&A at the end, if that's okay. So when you were developing the automated ICSI, what did you find was the most difficult obstacle you faced when you were working through the different steps?
Yeah. So for instance, here the most important part, I would say, is the AI system, the algorithm, and you have to do a new one when you use a different needle, a different species. Everything, just changing a little bit the bulb may result, or a different bulb system will result in different information. So the algorithms are very sensitive to all these changes. So this is probably the most difficult part. Every time that you introduce a little change, you have to redo the whole algorithm. So
different culture systems and culture media, you'd have to...
That we don't know. Definitely different species of eggs, and also different light sources, because that affects how AI works. So would it need to be adapted to each lab slightly to fit in with existing equipment, do you think? No, I don't think so. I'm just saying that since these were prototypes, every time that we change, let's say, a bulb from a different manufacturer, or the lenses were not aligned perfectly, and so on, you have to redo the whole AI system and retrain it again. Great. Well, that was a really fascinating innovation. Thanks very much. And
I'm sure people will put more questions in the Q&A which we'll cover at the end, if that's okay.
Sure.
Okay, it gives me pleasure to introduce the next speaker, and that's Karol Kujawa. He's working closely with IVF 2.0, looking at an exciting area. Karol serves as an extended realities lecturer at the Department of Game Research and Media at the Kazimierz Wielki University in Poland, and he has extensive experience as head of programs and projects in the IT industry. And he also co-owns the Unreal VR, a VR cafe in Poland, and he's going to talk to us about the minority report within the IVF lab. Thanks very much, Karol.
Thank you very much, Andrew. It's a pleasure to be here. So let's get this party started with a presentation, I suppose. Here we go. Is it visible?
Yes, it's fine, thanks.
Okay, awesome. So let's start with a little bit of an explanation why the minority report. So of course, it's connected to the Philip K. Dick's novel, which was got I don't remember exactly 50 or 60 years. It's something like that. So, and it's connected to the movie franchise, of course, with Tom Cruise. So why is my minority report? So it was the idea of having the data and being able to manipulate it in the real time using your gestures, your hands. So controlling, hand controlled, and the idea of being able to manipulate with the data with voice control, for example. So be inside of it, not just in front of our computers, our notebooks, smartphones, tablets, but be just inside of it. So there is I suppose, a lot of movies and books around it, but we'll focus on that one exactly. So what's the mixed reality? So mixed reality for us is a convergence of the real and virtual world to produce new environments and visualizations where physical and digital objects coexist in real time. So just as straight as it's only possible. So you've got the holographic overlay everywhere of data units at this point, and that's it. So what are the capabilities of mixed reality? So as I've mentioned, so it would be a seamless blend of physical and virtual worlds, interactions with both real and virtual worlds, and immersion. So this is the thing which I'd like to extend a little bit. So the immersion is very important here. Why is that? It's a little bit like a comparison of watching a Netflix movie on your, I don't know, 50, 60-inch TV at your home, comparing to going to the cinema and seeing it as a, for example, IMAX cinema as a totally wider range. So this is the thing which we call the immersion. So being able to get inside of your data, of 3D scans, of real data pulled directly from your servers, and giving it to directly in front of your eyes. It's something we call the immersion. But let's start with something which is widely known. So I would say the obsolete labs. I suppose that major labs still looks the same as I've listed here. So paper sheets, graph papers. So I know that the idea of manual sketching and handwriting your notes is good for your brain, I suppose, but still having lost those, it can be painful for the research you are doing. So notebook to record steps. So analog to digital manner. So all of the notes we've taken, we have handwritten to the paper sheets. At some point, are supposed to be transferred to the digital manner. So that's the huge potential for human error and mistakes. Miscommunication within the lab. So we know how it is. We know that when we are focused totally on the procedure and our colleagues walks to the laboratory, it could be a problem. It could be a total problem for you to focus on your work because of other people. So data access and security issues. So problems with having all of your data dispersed in variety of different servers, different software applications, nodes, not only analog paper nodes, but digital as well. Improper care of lab equipment. So when we don't know how to use it, for example, imagine that we've got a freshly hired employee and there is no proper instruction available how to handle the equipment. A lot of weird things can happen in the laboratory. So it happens still. We need to print out the PDF versions of instructions, manuals, so on and so forth, and those are flying around, and it could pose a huge risk for laboratory equipment. And of course, our beloved software, so the Excel spreadsheets, and how it looks right now. So from the perspective of a smart lab. So AI-enabled planning. So we, when planning the procedure, before we want just to plan it properly, we had to do it manually. Right now, it could be assisted as Microsoft Teams, for example. So another option, voice-controlled navigation. So there is no need to type in to look at the physical keyboard. We've got it. We've got voice assistants such as Siri, Google Assistant, Amazon, and so on and so forth. So that's just the case of implementation in the laboratory. So automatic sample processing. This is something which is existing, and previous panelists mentioned already. Smart inventory control. So I suppose that there is no need to use it and do it manually. So opening the Excel spreadsheet and count samples manually. It can be done automatically, and it is being done. Environmental monitoring. So when we've got the IoT-enabled device in our laboratory, we can send through the API all of the information to the system. Instrument interface, such as LIMS systems, ELN systems, real-time training. So the simple dashboard with all of the information pulled from the systems and presented in the simple labs. So all of this can be done and put into one device, which we call the mixed reality device. So goggles, headsets, a lot of different names. Just to mention a few available on the market right now. So the Microsoft HoloLens 2, Magic Leap 2, and Lynx R1. So those are the devices which are available already, and those are not the prototypes. These babies can really immerse you in the data and in the procedures you've been taking at this point using sheets of paper and your notebooks. So and seeing is believing. So this is one of the products, which is called the Holophore Labs. So what you are seeing right now is a recorded view from the first-person perspective. You've got a Microsoft HoloLens 2 goggles, so there's a semi-transparent display over your head, so you can see the real environment of your laboratory, of your desk, and additional layer of information. So this is the alias procedure displayed in a holographic manner. As you've seen a moment ago, there is an OCR option available, and as you can see, there are two ways of navigating through the holographic layer. One is a gesture control, so it's more like operating with a physical device. So that will be the first one, and the second one, as I've mentioned, is the voice navigation, so using the Cortana. About the mixed reality directly for the IVF market, I've been thinking about those three specific subjects as a first point. So patient education. So mixed reality can create an immersive and interactive experiences that allow patients to visualize and understand the complex of IVF process. So using the virtual, augmented, or mixed reality, patients can explore 3D models of reproductive organs, gametes, embryos, and the various stages of IVF, helping them grasp the steps involved so they can better comprehend the treatment. So a little bit more around it, interactive educational modules. So I was thinking about mixed reality as that can facilitate interactive educational models, that engage patients in a more dynamic and more memorable way. So patients can participate in virtual tutorials, quizzes, and interactive presentations tailored to their specific needs. This approach might encourage active learning, promotes retention of information, and allows patients to learn at their own pace. So next one is a medical training. So for example, surgical simulations. Mixed reality can provide realistic surgical simulations for training IVF specialists. So as I've said before, this can be helpful, for example, for the onboarding process for the new employees. Surgeons can practice procedures such as oxygen retrieval and embryo transfer in a virtual environment. So it's probably safer this way, replicating the condition and challenges that may encounter during actual surgeries. This allows for repetitive practice, skill refinement, and the opportunity to learn from mistakes without any risk to patients. So next one, from the perspective of a medical training in mixed reality, could be the collaborative training. So I believe that mixed reality enables collaborative training experiences where multiple trainees or medical professionals can interact and learn together. It is important when we have a dispersed team, so for some reason they cannot meet physically, so we can use mixed reality for that. So they can virtually participate in IVF procedures, discuss case studies, and exchange knowledge in real time. So this fosters teamwork, enhances communication skills, and encourages a multidisciplinary approach to IVF training. So imagine that you are sitting, for example, in Argentina in front of your laptop, and you're able to see everything what your colleague is doing, and he's in London right now. So you'll see everything as you've been there. So it's everything real time. Moreover, you can place all of the crucial information which would be displayed in front of your colleague's eye as a holographic layer. So for example, so the real-time guidance and feedback that can provide trainees with real-time guidance during IVF procedures. For instance, augmented reality overlays can offer step-by-step instructions. So, for example, as alias procedures, highlight critical anatomical landmarks and provide feedback on technique and performance. This immediate feedback loop allows trainees to correct errors, refine their skills, and improve their proficiency. From the perspective of procedure planning, so I was thinking about using the mixed reality for the IVF specialist to perform preoperative assessments in a virtual environment. So they can virtually explore the patient's reproductive organs and identify any abnormalities or challenges that might impact the IVF procedure. This helps in identifying potential complications and developing strategies to overcome them before the actual procedure. Next one, so the patient-specific treatment planning. So by incorporating the patient's medical imaging data, such as ultrasound or MRI scans, into the mixed reality environment, IVF specialists can precisely analyze the patient's reproductive organs and tailor the treatment plan accordingly. This individual approach increases the chances of success and minimizes potential complications. So, potential impact on IVF market. So, I believe that mixed reality has a huge potential, not only for the IVF market, but for all of the laboratories. And by integrating it into that, the technology can potentially revolutionize the work of clinical embryologists by enhancing their efficiency, accuracy, and training capabilities. Challenges, of course, as always, there are pros and cons of any new technology. So I will start with, I suppose, the major one. For me, it would be the cost of equipment and infrastructure. So mixed reality technology often requires specialized hardware and software. So it's not cheap, which can be costly to acquire and maintain. To address this challenge, organizations can consider partnering with technology providers or exploring leasing options to mitigate the upfront cost. Additionally, as technology advances, the cost of mixed reality equipment may decrease over time. And I believe that it will happen in the future. So another specific challenge when implemented mixed reality into the laboratory systems. So the integration with existing systems and workflows. So incorporating mixed reality into laboratory workflows may require integration. So from my experience, you need an API and few endpoints, and that's it. And all of the data can be pulled even from before mentioned the Excel spreadsheets. So it's essential to ensure seamless data exchange between these systems and the mixed reality platform. Collaborating with IT specialists and vendors can help identify and address any compatibility issues. So I believe that by implementing the mixed reality into the IVF laboratory, we are not supposed to create additional tool, because we've got so many of them existing already, and probably some of them are not even used on a daily basis. I believe that all of the data is inside of your servers, inside of your nodes. The only thing which we need is to put it in front of your eyes and eyes of your colleagues, of new employees. That's it. The last but not least, data security and privacy. Mixed reality platforms may involve the use of patient data, which raises concerns about data security and privacy. It's crucial to implement robust data protection measures, including encryption, access controls, and compliance with relevant regulations. So from the United States perspective, I suppose it would be the HIPAA. Conducting regular security audits and ensuring adherence to the best practices can help safeguard patient information. And seeing is believing, so it's not the future technology. So you will see by yourself.
At the official unveiling of our new Discovery Centre in Cambridge, we had the opportunity to show what science can do and to demonstrate how we're advancing our commitment to patients and the planet.
This realization of a vision we've had for the last number of years, building a state-of-the-art R&D center, we call it the Discovery Centre, the DISC, in the heart of Cambridge, surrounded by so many academic institutions that are the best in the world.
Opened by His Royal Highness, the Prince of Wales, we welcomed leading researchers from the scientific community, dignitaries and partners to see the pioneering research and technology we will be doing in the building.
Over 100 guests joined us to mark the occasion and experience firsthand how we're turning science into medicine and tackling some of the biggest healthcare challenges.
Esteemed guests and colleagues, I'm delighted to welcome you to celebrate this important milestone for AstraZeneca.
I don't think any company beats AstraZeneca in their commitments to science, to learning, to discovery.
Today really marks the culmination of many years of investment that has seen the transformation in our research and development capabilities.
I must say it has been absolutely fascinating to see some of the work that takes place inside this center, and to think that, supported by interactions across the city and beyond, it will ultimately enhance and save the lives of untold millions of people around the world in the years and decades to come.
We showcased some of the key scientific capabilities that underpin our R&D, investing in fundamental scientific understanding of disease to design the next generation of therapeutics, developing preclinical models that better predict the human body to improve patient experience and outcomes.
Our new R&D facility has been purposefully designed to be open and porous and to encourage interaction between our scientists and the surrounding scientific and medical community. The unveiling is just the start. The future of medicine is here.
Okay, so as you've seen, that was the. I haven't been paid by AZ to show that one for you. So there are a lot of use cases existing and a lot of proof of concept projects done already around not only the big pharma industry, but a lot of different laboratories as well. And mixed reality is a daily tool which is used over there. So just to step up to the conclusion, from my perspective, the mixed reality has the potential to revolutionize the IVF market, enhancing patient education, as I've mentioned, improving medical training and enabling better procedural planning. And please bear in mind that this technology is of today, not of the future. Thank you very much.
Thank you very much, Karol. That was absolutely fascinating, a real glimpse into the future. And I would certainly agree that there's huge potential for mixed reality and clearly huge benefits to real-time data capture within integrated systems and a shifting of this data into digital systems for far better insights. That makes it far more useful to the IVF practitioner and ultimately better for the patients, as you say, relating to their specific needs. We are going to open up the floor for questions from our audience to all of the speakers. But before doing so, I just have a very quick question for you, Karol, and thank you very much for sharing that case study relating to AstraZeneca's experience making the shift from human manual-based systems into using mixed reality systems, VR and AR. Was this a long and painful process for them, or was it relatively short and painless?
Well, to be honest, from the IT perspective, it was something I shouldn't say easy, but it was good. It went as planned, but the major restrictions which were spotted were with the human enablers. So I would say the problem is with the change in our brains. So we had some difficulties in convincing the scientists to stop working on Excel spreadsheets and move towards something which some of people still believe that it's something like a more the sci-fi device, the prototype device. But after a few training sessions, we've seen that no matter the age, no matter the experience, they've just started running around the laboratory using the holographic layers, which seems to be funny when you are looking at that from the external perspective. But yeah, so the problem with the change in our brains, I think that was the most important step.
That's really insightful. Thank you very much. And that echoes some of our own experience with similar systems relating to change management, but definitely worthwhile. Okay, so for now, I think we're going to open up the floor to general questions from our audience to all of the speakers. So be prepared. Dr. Drakeley, do you have a question that you'd like to kick off with, please?
Yeah, thank you very much. Questions from the audience, actually. So question, I guess, for Karol and Matt and Christine and Santiago, actually, and a couple about cost. So do we really think that all this high-tech stuff will make IVF cheaper, or will it make it more expensive and more out of reach? Where are the cost savings and efficiencies, do you think?
Obviously, we're doing this to be cheaper. As I said, one of the biggest limitations right now for people to have access to fertilization treatment is the cost. In the US, it's thought that only 10% of the people that are infertile can access it. So definitely, if you want to automate something, it has to result in savings in cost.
I agree. If I want to pitch in, I have to absolutely agree. There are entire algorithms saying that this does produce cost savings, all of them, across automation. If these savings are going to be passed along to clients, to patients, that's another question. But I think by automating and creating the algorithms and working with finances, which I think all of us are doing that, and we're doing an amazing job, by the way, we're doing our part. There will be an opportunity to decrease cost of IVF overall. If this is going to happen, I don't know. That's another ball game to me.
And I would echo that as well as having direct cost savings that are quite easily measurable, such as the savings in time and the efficiency gains that can be seen quite clearly by implementing electronic or automating systems. There's also the hidden costs. What's the cost of making a mistake? What's the personal tragedy? What's the cost to the patients? What's the cost to the hospital? They're costs that are very difficult to quantify, but we know from experience, we have enough experience now to know that the catastrophic mistakes that can occur have huge cost, which is almost immeasurable. So it makes a lot of sense to try to reduce some of that risk and mitigate it by introducing electronic or automated systems that remove some of the human factors that are responsible for the errors that are still occurring in IVF.
Okay, I'll ask another question about virtual reality then to Karol. Do you think it's the virtual reality controls are dexterous enough for clinical practice, or is it really just for data collection and recording?
I believe that based on my experience, so we've been introducing the mixed reality headsets in one of a company in Mexico. So the training from the level 0, I would say, for the company employees, I would say the change agents, as we call them, to the fully fluent manner of working with a mixed reality holograms, so all the holographic layers. It went within 15 or 17 minutes. So it's easy as it goes, trust me. So my kids are working with they are 7 and 10, and they are working with a mixed reality, with a snap of the fingers. So we can do that as well. So it's not that terrifying as it looks, trust me.
I have a question for Santiago. This is from the USA, from Alison Coates, who asks, how does it align the egg correctly for injection, i.e. PB at 12 or 6 p.m., for example?
Well, this is something that Nuno told me, Nuno Borges, who's one of the main ways, the principal author in the paper, that it does really, there is not too much evidence that the position of the polar body is so important. He, as you know, Nuno has done a lot of transfer of cytoplasm and so on, and the spindle, it's really tough. So even if you go through the spindle is going to move to the side. So really the position of the polar body is not very important. However, we're interested in the position on that position because then you have more perivitallin space around that. So we need to calculate for that. But really, we can inject in any position. It's going to be the same effect, we think.
Thank you very much.
Quite a few questions for Santiago, and I think obviously you're presenting your own work is very interesting. If you don't mind, I can carry on, Andrew. There's another question which I thought was interesting. I think with the video it wasn't clear, and someone's asking, Herman from USA is asking, was the suction for the holder and injection done technically or robotically, i.e. I think there is a lack of clarity around what steps were actually done by the robot. Mm-hmm. And I think it is a really interesting question because I'm sure a lot of people who followed your work have read the super complimentary editorial by Munne, Automated ICSI, and the tough road to automation, which is an editorial published after this initial data that Santi has presented today. And really what she does is talk about their achievements and how well the data was presented, but also tries to break down all the different steps, the micro steps within ICSI, and then talk about what is done, what has been done to date, manually, and then automated. And it's almost a way of, this is the checklist to full automation. And so it'll be interesting to talk about, I'm sure you've read that, Santiago, it'll be interesting to talk about, answer this question in the context of, I think, that overall view of what are the steps in ICSI, what can we automate, what can't we automate as yet, and what is important to automate, if not everything.
Yeah. So, well, we thought, for instance, here we use a pipette to hold the egg. Obviously, the holding pipette is not necessary, especially if you integrate that with let's say microfluidics to select the sperm. Then there are many systems or many patents that have been published showing that you can hold the egg through a micro device, right? So in one place you could have your egg, your sperm. They go through a serpentine, and you select the ones that are more viable. I mean, you have the zygote already that's doing that. So then you pick the sperm there, and you could hold the eggs with a holding pipette, right? That would be easy to do. In this case, in this prototype, basically, we just use a regular holding because we thought that that was not so interesting. The most interesting part was the injection, or the more difficult part. In the prototype that we use, we have to go down, pick up the egg, and on the holding, and that's it. And also, so we have to load the needles, and the needle it's loaded with a sperm one, and the other grabs the egg. After that, everything else is automated.
Okay, thanks. Denny Sakkas has also got a lot of questions. Obviously, Denny's not here to answer them, but we will be collating them and send them to Denny afterwards. But Christine's kindly offered to answer a couple of them. And the first one is from Charlene Elouf, who describes herself as an embryologist and fast car enthusiast. So the question is, do you think that the differential in metabolic states between the hatching trophoectoderm and those trophoectoderm cells still retained within the zona pellucida is mainly a reflection of the direct exposure to the culture media of the hatched cells?
Hi, Charlene. We adore you. Thank you for the question. This is actually a very good question. And yes, there is evidence on those studies that say that it is because of a mechanical touch from the trophoectoderm cells to the zona pellucida and without that. So it seems that after the trophoectoderm is no longer touching physically something that is the zona pellucida, they start stopping metabolizing for a certain period of time. So the short answer is yes, we have evidence on those studies that says that this is the correlation. If it is or not, we don't know, but this is the evidence that is there.
Thank you. And the second question from Pandyan Natarajan. Do you think there's a dialogue between the embryo and the endometrium, or is it a monologue embryo and with a sort of monologue embryo dictates the instructions to the endometrium?
According to the presentation and the data that was out there, we see suggestions in the evidence that there is a communication between both. It's not just a monologue. The embryo just doesn't go wherever. It feels like an implant. There is an attraction and there is kind of a communication between the two tissues that will lead or not to implantation.
Thanks, Christine.
Thank you.
Right, I have another question for Christine, actually, this time from California, from Moses Cesario, who asks, what's the timeline, in your opinion, for automated cryo becoming standard in the care of an embryology lab? Or the standard of care in an embryology lab?
Yeah, we have a guesstimate that in the next 5 years, we'll have cryobanks that cryopreserve oocytes automated. Overall, in IVF labs, I think we have to be a little bit more resilient and expect between 5 and 10 years. But I would like to hear the opinion of Dr. Munne on this, because he has such greater experience on all these processes of developing the automation. He's way ahead than me on all this.
So, well, thank you. We are going into trials for FDA approval. The trial probably is going to take a year or so, and then FDA bureaucracy hopefully is going to go much faster. But let's say 2 years. I think it will be in the market in 2 years. Here, for all these things that we're talking about, the handicap obviously is money and investment, and it takes. You mentioned a bunch of patents that were great, and then they didn't go anywhere because they needed money just to go through the FDA process. It's a huge investment.
Agreed. Thank you.
And could I just ask Christine a question, just slightly off topic? What about automated liquid nitrogen fillers? Is that something that's on the horizon as well?
That is very reliably established in Europe. It's something that exists for more than 10 years. I have worked with them. There are several brands right? And we know that some brands are going to fail and explode your tank. But there is a variety of products in Europe that are EU certified, and they work beautifully, and they have great customer service and installation. I have worked with a few of them, and I'm very satisfied with how reliable and how much cost efficient those are. So it's all here, it's not in the future. And a lot more IVF clinics in US especially are using that now, are making the transition. I think every other week I see a clinic making a transition to autofillers that are very reliable, and they converse very well with monitoring systems, the third-party monitoring system. So I'm a big proposer of that.
Thank you.
There are a few questions for Christine. I'd like to ask one from Dr. Karen Savaya. She talks about, I mean, we've touched on this, the main things that you talked about, the advantages of automated vitrification, but she asks about the data for accuracy and precision, and you've given us some of the papers, but she also says, what is the data on implantation rate, live birth rates? And actually going on from that, and we don't have much time, but it's really, how do you control for that, given all the other rate limiting steps? How do you standardize all the other steps so you can show the success rates of automated vitrification versus sort of manual vitrification?
This is an excellent question. We have a lot of very standardized protocols that randomize as much as possible the data that we're going to use for these studies. And there's a lot already out there, but not in US. In US, hopefully, if everything goes well, we're going to start testing maybe next month or in the next couple months with samples that are very precisely randomized. So we know statistically that only the vitrification is the only variable or significantly variable data that we're collecting in there that is going to have an influence or an impact on the clinical data that we're going to take out of it. It's coming. There is a lot already out there on those papers that I put in the talk, but a lot more from US is coming in the next few months. Just pay attention on the papers, pay attention on how it was randomized and if it was validated properly. And I'll take any questions you guys have further.
Thank you. That was brilliant. Thank you. Do we have time for one more question, or are we wrapping up?
Matt, any final question from you?
Probably for Karol. You've spoken about the potential benefits to the IVF industry. When are you going to start working within the IVF industry?
Okay, so I suppose that it would be good to mention that IVF is currently looking into the proof of concept use case. So that's the first laboratory can implement the mixed reality and see how it goes. So I would say that it would be more like a proof of concept and a proof of value project. So after a few months, we've got all of the KPIs in Excel spreadsheets, of course, filled in. So we can say that moving from the current state to the mixed reality state, we achieved, for example, 30 minutes per one scientist per day of a faster, for example, procedure execution. We've achieved, 17% human errors less in comparing to the previous states, and so on and so forth. So be patient, please, and we'll be sharing more details soon, I suppose.
Well, that's fantastic. Thanks very much, everybody. So we would like to thank all who took part in the webinar, certainly the contributors to the questions, but also especially my co-host, Stephanie Kuku, Matt Pettit, and presenters Denny Sakkas, Christine Allen, Santiago Munne, and Karol Kujawa. Thanks for joining.
Thank you, Andrew. We'd also like to thank our team from i3, the organizing committee of this initiative, who are Jacques Cohen, Peter Nagy, Marianne Savites, Thomas Elliott, Fran Farley, Giles Palmer, and Violetta Sura. And also thank you to IVF 2.0 for sponsoring this session. We want to stay close to you, tell you what topics you want, tell us what topics you want here. And remember, we have podcasts and webinars, all uploaded free on the IVF meeting website, ivfmeeting.com. Thank you.
Thank you, Stephanie, and thank you once again to all of our speakers for what proved to be a fascinating and thought-provoking glimpse into the future of IVF as we tentatively but boldly cross over from manual human practices to electronic or even automated systems that have huge potential to be able to mitigate the risks that lead to errors, to drive efficiencies, and hopefully to improve clinical outcomes. And as Christine said so eloquently, let's take a breath and open our minds. So keep your ideas coming in for new and interesting topics into the future. All of us here will be attending the ASHRA meeting coming up, and we'd love to hear from you if you're going, and we'd also love to meet you there. So keep in touch. Thanks very much.
Thank you.
Bye, everyone.
Bye.